Welcome to BioPharm International's podcast, SARS-CoV-2 Molecular Testing, From Research to Wastewater Surveillance. This podcast is brought to you by Thermo Fisher Scientific, the world leader in serving science-enabling customers to make the world healthier, cleaner, and safer. To find out more, please go to thermoFisher.com/sampleprep-SARS-CoV-2. And now, here's your host for this podcast, the contributing editor for BioPharm, Cindy Dubin.
Hello, everyone. This is Cindy Dubin, contributing editor for BioPharm International, and I'm here with Alexander Sasha Vlasov, Senior Manager of Research and Development at Thermo Fisher Scientific. Thank you for being here today, Sasha.
Thank you for having me.
So there are several approaches for SARS-CoV-2 testing, such as antigen, antibody, and qPCR. Can you tell us more about the different options and how they compare?
There are three different testing options for SARS-CoV-2: detection of viral proteins or antigens, detection of viral nucleic acid by PCR, and detection of virus-specific antibodies in the blood. Antigen and PCR tests determine whether an individual is currently infected with coronavirus, while an antibody test can determine if the subject was infected more than 2 to 4 weeks prior. When it comes to COVID clinical testing, the most widely used approach at the moment is qRT-PCR. The starting material is coronavirus RNA, purified typically from the swab sample, which is converted to DNA sequence by reverse transcription, and then quantitative PCR is performed to amplify the material and determine if virus is present or not. The process typically takes 2 to 4 hours from sample to answer, but it is the best test option, capable of detecting 100 to 1,000 viral copies per milliliter. PCR approach is extremely sensitive and specific. In response to COVID outbreak, Thermo Fisher Scientific developed a new multiplex real-time RT-PCR diagnostic kit to enable clinical and public healthcare laboratories to quickly diagnose COVID-19. The Applied Biosystems StackPath COVID-19 kit is a fast, highly sensitive multiplex diagnostic solution that contains both the assays and controls needed for the real-time PCR detection of RNA from the SARS-CoV-2 virus. And going back to antigen testing, which is detection of viral proteins, it can be as fast as 15 minutes, and many test formats are very user-friendly, which is definitely attractive. But keep in mind, there is no amplification step for protein-based detection, and as a consequence, test sensitivity is dramatically lower versus PCR, usually over 10 million viral copies per milliliter.
I understand that some of the tests are based on nasopharyngeal swabs, but there have been reports about the use of saliva as a new sample type. Can you tell us more about that?
Nasopharyngeal swabs were almost exclusively used in the early days of COVID testing, and they are currently still #1 most popular sample type for clinical testing. Standard sample collection kits for SARS-CoV-2 testing use nasopharyngeal swabs and storage tubes containing either universal transport medium or viral transport medium. Despite many years of successful use, nasopharyngeal swabs have a very important disadvantage. They can be collected only by trained specialists, and the process is rather invasive and painful. Other sample types are oropharyngeal, nasal, and mid-turbinate swabs, nasopharyngeal aspirate, bowel, sputum, and saliva, and there are some other rarely used sample types. Overall, it's a pretty large number of sample types collected and used by different medical facilities depending on their preference. Saliva, nasal, and oral swabs became very popular due to their ease of collection. They're perfect for at-home collection format or rapid screening surveillance in the field when medical specialists are not available for assistance. There are pros and cons for each sample type. For instance, saliva is completely non-invasive and easy to harvest sample, extremely attractive. But for some people who have dry mouth, it takes up to 20 minutes to collect 2 milliliters of saliva. So it's extremely challenging in some settings. And also it is very important to avoid eating and drinking for about 30 minutes prior to harvesting the sample. So depending on the situation, some organizations prefer to deal with saliva and some others switch to nasal swabs or oral swabs for fast testing.
So Sasha, we've discussed different options and solutions for SARS-CoV-2 testing, but recently there was an exciting new development with wastewater surveillance. Can you tell us more about that?
SARS-CoV-2 is reliably detected in the stool of most COVID patients, beginning at the pretty early stage of infection and for several subsequent weeks. And based on these findings, wastewater or sewage surveillance became an extremely useful tool for monitoring the regional spread of SARS-CoV-2 and other viral and bacterial pathogens. Hundreds of organizations worldwide are successfully using wastewater testing for surveillance and early catch cases to prevent COVID-19 clusters. Last month, officials in Britain announced testing at 44 water treatment facilities. The Netherlands has been collecting samples at 300 sewage treatment plants. In the United States, over 200 water facilities across 40 states are being tested, and the number is constantly growing. The CDC and the US Department of Health and Human Services initiated the National Wastewater Surveillance System in response to the COVID-19 pandemic. Many universities have organized wastewater COVID testing programs. Notre Dame, UCSD, Syracuse University, New Jersey Institute of Technology, Utah State University, and other. For instance, University of Arizona is testing sewage for all 20 residence halls. When a wastewater sample from one dorm came positive a few weeks ago, there was an announcement. The school quickly responded, tested all 311 people who lived and worked there, and found 2 asymptomatic students who tested positive. They were quickly quarantined. So the benefit of testing the water is that we get a snapshot into what is happening on campus and can quarantine even before a student becomes asymptomatic. Wastewater testing approach is inexpensive, fast and simple, not requiring sample collection from the individuals. So there's no face-to-face contact, and the entire workflow is pretty robust and carefully optimized by several institutions using it right now. So overall, early work has shown that wastewater surveillance at different levels of operations, such as dorms or accommodations at the university or the entire city, could reliably detect SARS-CoV-2 before it became widespread in population, and also to monitor its spread at the city level.
That's really interesting. Thank you for that explanation. Please correct me if I'm wrong, but quantitative PCR seems to be the most highly adopted technique for SARS-CoV-2 testing. What more can you explain about this workflow, such as the types of kits and instruments used?
Yes, indeed, qPCR is currently the leading approach for SARS-CoV-2 testing. For quantitative PCR, we'll focus on description of the researcher's only solutions for SARS-CoV-2 testing. The workflow involves several steps: sample collection, sample preparation, real-time PCR, and data analysis. SARS-CoV-2 RNA can be purified quickly and efficiently from samples using the MagMax viral pathogen nucleic acid isolation kit. And we have 2 versions of this kit. The product is magnetic bead based, which enables high throughput sample processing using either magnetic stands or specialized purification instruments. MagMax viral pathogen kits can be, for instance, automated on Kingfisher Flex system for fast, easy, and effective high throughput nucleic acid isolation. The Kingfisher Flex instrument is a versatile benchtop instrument for processing up to 96 samples per run. For SARS-CoV-2 pathogen detection, there are several workflow options from manual to fully automated scenarios, depending primarily on the desired throughput. That means number of samples processed per day in this particular laboratory. Semi-automated workflow with Kingfisher Flex instrument and magnetic beads is one of the most widely used workflows for medium throughput SARS-CoV-2 testing. The viral RNA isolation protocol takes only 25 minutes. Hands-on time is dramatically reduced compared to the manual approach. And the typical lab using 1 to 4 Kingfisher Flex instruments can comfortably process 1,000 to 4,000 samples per day. Once viral RNA is extracted from the samples, the next step is qRT-PCR test, which determines the levels of the viral RNA and generates a primary analysis. Quantitative RT-PCR can be performed using applied by cesium Stackman assays on qPCR research instruments such as applied by System 7500 series or Quant Studio systems. The software then calculates and analyzes CT values of the controls and specimens of each run, and in general, the entire SARS-CoV-2 testing workflow takes 3 to 4 hours from receiving the sample to completion of data analysis.
Thank you so much for this informative overview today, Sasha. We truly appreciate you being here.
Thank you so much.
This has been Cindy Dubin, contributing editor for BioPharm International. Thanks to all for listening.
You've been listening to BioPharm International's podcast, SARS-CoV-2 Molecular Testing, From Research to Wastewater Surveillance. This podcast is brought to you by Thermo Fisher Scientific, the world leader in serving science-enabling customers to make the world healthier, cleaner, and safer. To find out more, please go to thermofisher.com/sampleprep SARS-CoV-2.